Oh, there is a clock. Oh, we love that. Yeah, just in case I'm too locked in. But it's good to know that I have a clock.
Time though, so don't.
I'll be like, oh my God, it's 10 already. I've been talking for so long. Yeah, you good. I'm gonna black out. No, I think it'll be okay. Yeah. Be like, no one knows about lipids. Okay. Yeah, that's fine. I was like, who would be late? Okay, great. What is our time at? Oh. Okay, great. Wow, and Emily Parker's here. That's how we know we have to get started. Should we get started? Oh my God, and Jane's here. Perfect timing. I think it's exactly 9.
Okay. All right, everybody. Welcome to the defense of Maya. And apparently, this is supposed to mimic me. Maya came to us from Cal State Polytechnic University in San Luis Obispo. She worked there with Lars Demonic, who was also the OSU product. And during her honors research, she got to watch mussels grow. I think she's done something different here, and we have a guest producer, Sarah Selke, who is a fellow graduate student who is going to get her good stuff.
Do you want my mic? No. Okay, I'll just stand next to you.
Okay. Hello. I wanted to introduce Maya and some things she's been up to while we've been here at OSU together. So the first is that Maya, throughout her time at OSU, has been a really incredible mentor and teacher. She served as the mentor TA for the 22X series last year, which is a huge undertaking, and has really dedicated a lot of her time to caring for undergrads and mentoring us fellow grad students in the field, and teaching us how to have fun while we do our research as well. She's also been a really important organizer. She was the president of our grad student union and served on the executive council for countless years. So I can't remember the actual number. And so she's really fought hard for workers' rights and the rights of all grad students at OSU, and has made a huge impact on our community here. And then Maya is also, I will cry, a really important friend for all of us in the lab. That's all I have to say. That's all I can say. So we're excited to see what she does next. We're all super proud of her.
Wow. Wow, that wasn't nearly as embarrassing as you told me it was going to be. Okay, yeah, hi everyone. I'm so excited to be here today to share with you what I've been up to for the past 6 years. And all of my thesis research that I'm going to be talking about today deals with the high intertidal here in Oregon, and more specifically looks at how different grazers and thermal stress might be altering this nearshore community by us. So first I'll start with a little bit of introductory information. So for those of you that don't know, the intertidal is the region where the land meets the sea. It's this area where the tide comes up and covers the rocks some of the time, and then some of the time it goes out and exposes these same organisms to aerial conditions. And what makes the intertidal a really interesting place to study ecologically is that these organisms are having to go through a whole bunch of stress throughout a single day as the tide comes in and out and changes the temperature, changes the amount of desiccation stress they're under, how dry it is, and even things like their access to their food sources. So it means that we can study different ecological processes in the intertidal and then apply those to more not stressful communities and see how they might respond to increases in stress, especially with regard to climate change. The other thing that's really cool about the intertidal is that because of the way the tide works, you get these really distinct vertical zones of different biological communities, all of which are kind of adapted to a different intertidal life. So we have the low, mid, and then the high zone, which is what I study. And as you might expect from the name, the high zone, it is the highest vertical point where the tide still comes in and regularly covers the rocks throughout the day. And that also means that it is the hottest and driest region of the intertidal because of that, because it is exposed to the air the most out of these three regions. So let me orient you a little bit to Oregon's high zone. Imagine you're out on the rocks with me, out on the coast in Newport or wherever you want to be today. There are a whole host of different organisms that are out in the high zone, but I'm going to focus on a few major functional groups for my talk today. First of all, there are a bunch of invertebrates that live in the high zone, most of which are barnacles, which you can see a really good example of here on the left-hand side of this picture. We also have a lot of temporary communities that we refer to as microalgal or biofilm communities. And these are communities that crop up as the tide comes in, makes the rock nice and moist for them to create these microscopic communities full of diatoms, phytoplankton, things like that. And these are a really important food source for a lot of organisms that I'll be talking about today. We also have more permanent algal residents in the high zone that are divided into two major categories. The first of these is what we call fucoid algae, and this is a type of brown algae, so related to kelp out on the coast. And you can see some really good examples of that in this photo here. And then we also have our red algae, which you can also see mostly up in the top of this picture here as well. So there we go. We also have a whole bunch of mobile organisms that exist in the high zone, so those that are able to move around more throughout the community. And I'm going to be focusing on my three superstars for today, which are the major limpet species that we have in the high zone. Limpets are a type of mollusk. They are a type of snail. A molluscan grazer is what we call them. So they are herbivores. And all of the ones that we have in Oregon in the high zone belong to the genus Laudia. The first of these is Laudia digitalis here on the left. And if you are out in the high zone, this is probably the limpet that you are going to see. It is incredibly abundant, and it also tends to hang out in these little groups on vertical surfaces with each other. It's also a medium-sized species compared to the other two. Next up, we have Laudia pelta. This one is less abundant than digitalis. It likes to hang out also in the mid-zone, so you don't see it quite as much in the high zone, but it is still present, and it is also the largest of the three species. And then finally, we have a bit of a mystery species, as I like to call it, in Laudia paradigitalis. And the reason it's a bit of a mystery is because it's relatively understudied compared to the other two species. And this is partially because it is less abundant than the other two and is also the smallest. So there's kind of this assumption that it is ecologically less relevant, but we don't actually know that for sure. We don't have as much research on it as we do the other two. So as I mentioned, there are a whole bunch of different macroalgae that live in the high zone. I'm going to be focusing on 5 of the most common ones today that are in Oregon's high zone. 2 of these are those fucoid algae that I was talking about, Fucus disticus and Pelvetiopsis limitata. I'll be referring to all of them by their genus names just for ease today. And then we also have 3 red algae. We have Mastocarpus papillatus, Maziella parksii, and then we have Endocladia muricata, which is the only algae I'll be talking about today that does not form a canopy. So you can see that these other ones form these really beautiful blades that create a canopy just like a tree would in a forest. Endocladia is the only one that doesn't do that. Okay, so now that we have an understanding of these different functional groups in the community, let's talk about how they actually interact with each other. So the first thing that we have a pretty solid understanding of is that these limpets like to graze on microalgae. They like to graze on these temporary communities that show up on the rocks. And again, we have a lot of really good research showing this. This is their preferred food source. This is what they sustain themselves on in the high zone. We also know that there are some indirect relationships between limpets and space occupiers in the high zone, and I'm going to be highlighting barnacles as kind of an example of that for today. And the reason they have this negative indirect relationship is because as they're moving around or grazing on the rocks, limpets are really well adhered to the surface, and so they will literally bulldoze whatever is in front of them, dislodging them from the rock, allowing other things to potentially colonize that newly bare space. We have a pretty good understanding that this happens, but not necessarily how often and how strong that relationship is. And then finally, we have a bit of a mysterious relationship between limpets and macroalgae in the high zone. You would assume, because they're grazers and this is algae, that they would have a negative relationship or that they are feeding on these macroalgae. We don't actually know if that is the case, and we certainly don't know if it is strong enough to have any community level impacts. So they might be feeding on them a little bit, but not enough to really influence the amount of macroalgae, which species exist in the environment, that sort of thing. We also don't know if it's necessarily always a negative relationship because, like I mentioned, there is this negative relationship to other space occupiers that compete with this macroalgae. And so some studies have shown that limpet presence is actually beneficial to some macroalgae because they are clearing out space for them to colonize on the rocks. And so a lot of my research I'm going to be talking about today is kind of unraveling what this mysterious relationship looks like. One other really key component of my thesis research is thermal stress caused by climate change. So the intertidal, like a lot of environments across the globe, is dealing not only with an increase in average temperatures overall, but also an increase in what we call acute thermal events or heat waves. So you can see from this graph here on the right from Oliver et al. 2019 that the x-axis is years projected out to 2100. The y-axis is the total number of days that can be considered a marine heat wave day. And then these two different colored lines represent a projection of a more conservative climate scenario in orange and a more intense climate scenario in red. Regardless of which one you look at, you can see that we have already experienced a pretty exponential increase, and that is going to continue as the years go on. So these organisms are really going to have to deal with sudden extreme climate events more often as time goes on. So what do we know about how they might be responding to this stress? Well, we know that limpets actually may be relatively unaffected by thermal stress, at least as we currently understand it. There's been a lot of research done that shows that they are not really anywhere near what we consider their thermal maximum, right? So the point at which they start to die more often, have physiological effects, that sort of thing. They're kind of chilling up there, right? So they may be unaffected by these thermal stress events, whereas macroalgae, we know, are not nearly as thermally tolerant. And not only are they not as thermally tolerant, they also have negative effects that make them more susceptible to grazing. So some of them create chemical defenses, and those are reduced by being exposed to heat stress. They also can have a reduction in the actual toughness of their physical blades, both of which make it easier for a grazer to come in and feed on them, maybe if they weren't able to before as effectively. And the final component of this is that thermal stress usually also comes with desiccation stress. And the combination of those two things may be reducing the abundance of limpets' primary food source in microalgae. So we're facing a scenario where limpets may be relatively unaffected by heat stress. They may be losing their primary food source, and macroalgae may open up a new opportunity for them to feed on as they lose their main food source. So before we get into all the climate change of it all, first I had to really understand how limpets are currently affecting this high zone community in Oregon. So to remind you of our food web here, the main thing I wanted to focus on was this mysterious relationship of how limpets as a functional group are affecting the high intertidal, and really especially getting into how they're affecting macroalgal abundance, since that's kind of more of an unknown for us. And also, I wanted to see how each of these three major limpet species are driving whatever response we see for limpets. So is it mainly driven by one species? Is it all of them combined? What's going on there? To do this, I set up a field experiment at 3 different sites along the Oregon coast that will look very familiar to some of you. And at each of these field sites, I set up a whole bunch of 15 by 15 centimeter plots, some of which had a copper boundary around them. Copper is a very effective deterrent for certain invertebrates, limpets included, so they don't like to cross over these boundaries. So this allows me to keep them out or keep them in, whatever I wanted to do, without interfering with other mobile organisms in the environment, which is really nice. These plots were scraped initially to remove any biomass that existed there, and this allowed me to kind of track the community from an initial recruitment stage into a more mature form. And at each of these sites, I had 5 replicates of my setup, which looks something like this. So for each replicate, I had 6 treatments. I had a control, which other than the initial scraping, I did not touch. So limpets of all 3 species were allowed to go in or out as they please. I also had a copper control, and the only difference between that and the other one is that it had an incomplete copper boundary, just to make sure I wasn't seeing any sort of strange, unexpected effects of the copper just existing in the environment. I also had a complete grazer exclusion that you can see here in the bottom right, and that, like you would expect, means that none of the grazers were allowed in. There was a complete copper boundary, and none of the 3 species were in that community to influence how it matured. And then finally, I had 3 different, what I was calling single species treatments, where only one of these 3 major limpet species was allowed into the plots. And I made sure that they were at a abundance similar to what I would be seeing under natural field conditions. So the digitalis plots had more than the pelta and paradigitalis plots per individual. Once I had all of that set up, I measured community structure using photographs of my field plots, which I took 1 to 2 times per month as field conditions allow. They often did not allow me in the winter, and that's okay. And this experiment was put in place from early 2022 to late 2024, so about 2.5 years of data that I was working with. The first way I want to display that data to you is through an NMDS graph, non-metric multidimensional scaling, for those of you that want to know the abbreviation. And all this means is that I've taken all of my really complicated community data with all these different species response variables, and I've kind of smushed them down into 2 axes so I can actually look at how the community is changing over time. Each point that you'll see on this graph represents 1 plot at 1 time point. And then you'll also see that they are color-coded by treatment over here, which you can take a look at the little cheat sheet if you need to. And then there will be an oval to show where the average clustering of each treatment is. And if I do all that, it looks a little something like this. So the main thing I noticed when I was analyzing this is that the treatment groups did differ from one another. So there was a difference versus the complete limpet exclusion, the controls, the single species. Something was changing there. More specifically, I have a few patterns I want to point out to you. The first of which is that the controls did not differ from each other, so I wasn't seeing any sort of unexpected effects from the copper, which is really good. I did, however, see that both of these controls differed from all the other plots. So the limpets, the plots where the limpets were allowed in at their regular capacity, all 3 species, were growing a different community in the plots compared to my single species and my complete exclusion. If we get into more of a specific per species response, there's something really interesting that happens. So the Ladia digitalis plots, which you can see in blue here, differed from the complete limpet exclusion, which means that whatever community was growing was not necessarily just the community that grows when you have a lower number of limpets or no limpets at all. Whereas the same cannot be said for the pelta and paradigitalis treatments, which were not statistically different from the complete limpet exclusion. It was as if those individuals weren't really there. They weren't influencing the community to a degree to develop a different community. Yes, great. So what did this community actually look like? To show you a few examples of this, I have 4 different bar charts here. On the x-axis is the different treatments, and then on the y-axis of each of these charts is the average coverage of a different functional group. And you'll notice that the y-axis range for these are a little different for each one. So when we look at this, we also see something that is a little bit interesting compared to what you might expect from previous research. The first of which is that the barnacle coverage and the coverage of invertebrates generally did not differ at all between the treatments. So despite the fact that we've seen in previous research that there is this negative indirect relationship between limpets and these space occupiers, I wasn't actually seeing that in my plots. So there is a potential that it is not nearly as strong as we were expecting it to be, at least in this particular system. We did see that the microalgae coverage in the control plots where all 3 limpets were allowed in was significantly lower than those for the single species and complete exclusion. So they were eating them at such a rate that they were really significantly reducing the abundance of that food source, which makes sense. And then finally, for these two, I have 2 different metrics of macroalgal coverage. I have their holdfast coverage and then that canopy coverage that I was discussing earlier. And while there is a visual trend of the controls and the digitalis plots being lower than the other ones, this wasn't a significant pattern. So I can't say for certain that they were actually having a true negative effect on macroalgae. So to talk about these single species effects a little bit more, something interesting that I want to point out is that when you look at the digitalis bars on each of these graphs, excluding the barnacle treatment, you can see that it is kind of almost at this middle ground for a lot of these compared to the controls and compared to the other limpet manipulations. So digitalis is clearly serving as the primary driver of community change in this environment. So to sum up, how did we see limpets as a functional group affecting this community? We saw that limpets are affecting community composition, but they didn't have necessarily the effect that we might have been expecting. So there were negative effects on algal presence, especially microalgae, but no effect on invertebrates, which is interesting. We also saw when we look at specific species coverage that digitalis, as the most abundant species, was also the most influential. Whereas the other 2 species did not have communities that differed from plots without limpets. It was like they were at least ecologically, functionally not there. So now, armed with my advanced understanding of this community in its current state, I really wanted to dig into how limpets specifically are grazing on this macroalgae, if they're even capable of it, and whether heat stress would be altering these relationships. So again, to remind you of our food web here, we may have seen that there were no major effects at a community level in chapter 1, but that doesn't necessarily mean that we understand how things are operating at an individual level. Are limpets even capable of feeding on these macroalgae? Is there species level differences? And importantly, how does heat change this? So there is a potential that maybe nothing is happening right now, but that might be different as climate change worsens. So for this chapter, I was really testing the effects of 4 different things. I was testing the effects of limpet species. So we had our 3 major limpet species involved in this. And then I was also testing the effect of macroalgal species. So we have Fucus, Pelvetiopsis, Mastocarpus, and Mozziella are 4 canopy-forming macroalgae that I'll be focusing on for this. And then I also was testing the effects of algal heat stress and limpet heat stress. So how is heat stress on both of these different trophic levels affecting this relationship? To do this, I set up a lab experiment at Hatfield's experimental seawater facility where I collected a whole bunch of specimens from Yachats Beach in Oregon, and I placed them in small tanks with a simulated high and low tide cycle, high tide being 16 hours, low tide being 8 hours. This was a very advanced setup where I would take the tanks and take them out of the water and then put them back in the water after 8 hours. But it worked. And then I had 5 limpet individuals per tank. And when you put all of that together with all of my explanatory variables, you get a very complicated setup that looks like this on the right. So to explain what you're looking at here, each of these blue rectangles is one of my flow-through seawater tables that you saw in that previous picture. One of them was receiving stressed algae specimens, and I'll explain how I did that in a second. The other one was receiving unstressed algae specimens. And then in each of these tanks, represented by the black circles, I had my 5 limpet individuals in brown. Each tank was a specific combination of 1 limpet species and 1 type of limpet heat stress. And I had 3 levels of limpet heat stress that I was working with. The first of these is what I was calling the acute heat stress treatment, and this was meant to simulate a heat wave scenario, essentially. So these limpets were exposed to 25 to 30 degrees Celsius air temperatures just during the low tide before a feeding trial took place. And this temperature I got by taking the average maximum summer temperatures along the Oregon coast in the past 10 years. So this is an extreme scenario for them, but one that they could realistically be experiencing already. I also had a chronic temperature stress treatment. These limpets were experiencing more of a general average increase based on climate change scenarios. So they were exposed to 18.5 degrees Celsius for every low tide for 2 weeks prior to a feeding trial and during the feeding trial itself. And this is 3 degrees Celsius higher than the current average summer temperatures in Oregon. And then the control treatment, we're just experiencing ambient air conditions. For my algal heat stress, again, I had just stressed or unstressed samples, and my stress samples experienced something really similar to the acute heat stress limpets, where they had exposure to 25 to 30 degrees Celsius air temperatures just for that low tide before I put them in the tanks for the feeding trials. For those trials, I starved the limpets for 48 hours beforehand to make sure they were hungry, but not so hungry that they would be eating whatever I put in front of them. And they were each exposed to blade samples of all 4 algal species at once. So they were able to make a choice between these 4 macroalgae, which one they would prefer feeding on. Those algal samples remained in the tanks for 16 hours, which is one high tide sequence. And before and after, I measured their wet weight, their length, and their width. And then I repeated this entire experiment 4 times with different limpets in the tanks each time. And I'm going to take a drink of water. Okay, so once I had all these data, I calculated 3 different measurements of percentage change for the algae. I had the percent change in weight, which was just the before and after of their wet weight. I had a percent change in their surface area, where I just did a simple length times width calculation. And then I also had a percent change in what I was calling size, or a size index, which was length plus width. And I did both of these because these weren't necessarily rectangular samples, and I wanted to make sure that I was accounting for that and having 2 different options, just in case they weren't consistent with each other, which they were not. So you will be seeing all 3 of these metrics on the graph as we go forward, because all of them are kind of behaving in different ways throughout this experiment. The first thing I want to show you is a bar chart showing the different limpet species on the x-axis, and then all of these charts will have a similar y-axis, which is the average percent change for a given group. A positive value means a gain in whatever metric we're looking at, and then a negative value is a loss. So how did limpet species influence the grazing behavior? It did not. So the limpets did not care. In terms of species level, they had no difference in their feeding behavior. They were all grazing at similar rates, which was really interesting considering what we saw in chapter 1, where digitalis was really the dominant species. Then, if we're thinking about their temperature treatment, we had acute, chronic, and control. I had this whole fancy setup for them. How are they going to respond to that? They do not care what I did. They were not influenced by the temperature treatment at all, which makes sense to some extent. Like I was saying, they are very thermally tolerant. But seemingly that includes both a heat wave scenario and this lesser sort of gradual climate change scenario. Neither of these influenced their grazing rates, which was really interesting to see. So now, how is macroalgal species and the macroalgal temperature treatment going to impact grazing rate? Turns out this actually does influence stuff. And this looks a little complicated, but I'll talk us through this real fast. So I saw that the percent change in weight and the percent change in size were differing by algal species. So these limpets did have a preference in which type of macroalgae they were feeding on in these tanks. Specifically, I saw that Mastocarpus, this red algae right here, was not grazed on regardless of conditions. Limpets really didn't seem to prefer that one as a food source. 2 of the other species, on the other hand, Pelvisiopsis and Mozziella, were consistently grazed under any conditions, but when they were temperature stressed, which you can see here represented by these red bars, they were grazed on at a higher rate than they were in the unstressed treatment. So heat stress was having this negative effect that we might expect based on previous research. And then we also can see that Fucus was behaving very strangely for me, specifically with its weight values. So it was being grazed in the unstressed treatment, which you can see here by this blue bar. But in the stress treatment, those of you that know anything about the physiology of Fucus might be able to guess what was happening here. Fucus is a very hollow algae compared to the others. It's usually filled with air and water. And when it gets heat stressed, turns out it deflates like a balloon, which I did not realize until after this happened. And then you put it back in the water and it reinflates. So I was seeing really crazy gains in weight for this species, which really freaked me out when I was first looking at the data. So based on weight alone, we can't really determine how it was being grazed per se, but we can say that it was being grazed at least in this unstressed treatment. So to sum it up real fast for you, which limpet species can feed on which macroalgal species? It turns out that all limpet species seem to feed at equal rates, at least when they're isolated from each other in these tanks. We see that Pelvetiopsis and Masiella are consistently grazed regardless of conditions, and that Mastocarpus is seemingly not preferred by these limpets usually. In terms of the effect of temperature, we saw that limpets can withstand both acute and chronic heat stress, at least that is similar to conditions they might currently be experiencing in the field. But that temperature-stressed algae, which was stressed under the same conditions, will be grazed more heavily by these limpets. So now that I had this understanding of both the community structure from chapter 1 and their feeding behavior and how that relates to climate change in chapter 2, I wanted to take all of that and combine it together into one final field project where I would see how limpets and heat stress are affecting this high zone community as a whole. So to take you back to the food web one last time, I was looking broadly at the effects of the high intertidal community, the effect that heat stress would have on the high intertidal community. And specifically, I was just looking at that acute heat stress, so those heat wave scenarios in this chapter. But then more specifically, I wanted to see how are limpets potentially modifying this response? How are they maybe driving this response in the community that we might see based on their heat stress scenarios? So, like I mentioned, I went back out into the field to Fogarty Creek for this project, where I set up something pretty similar to what I did in chapter 1. This time I had 7 replicates, but I still had these 15 by 15 centimeter plots with these copper boundaries to include or exclude my limpets. But this time I was also manually applying heat stress to certain experimental plots using my very fancy setup that you can see here. And this is just a PVC square that has a aluminum foil pie tin in the center with an electric hand warmer inside. And that sort of served as a heat lamp that I could set up in the field to create this acute heat stress scenario. And I would take these out every 1 to 2 weeks to reapply about 1 hour of heat stress. So a relatively short but cyclical heat stress that they were experiencing. For this experiment, I had 4 treatments this time. I had a control, which in this experiment, I was not clearing the plots initially. So this was just the existing community that was already there. I just set up 4 lags and called it a day. I didn't touch anything with the controls after that. I also had a limpet exclusion plot. So once again, nothing was scraped or anything like that. I just had an existing community that I painted a copper boundary around to keep limpets out. And if there were limpets in the plots to begin with, I went through and removed all of them during installation. I also had heat stress plots where limpets were allowed to come and go as they please, but they were experiencing that acute heat stress every 1 to 2 weeks. And then finally, I had a combination of the two where the plots were experiencing both acute heat stress and also limpet exclusion. And these plots were measured every time I went out every 1 to 2 weeks from May to August of 2025. So once again, I want to start off by taking a look at an NMDS to understand generally how the community is behaving. And you can see here on the right a quick cheat sheet of what these treatments are and which color corresponds to which one. As a reminder, 1 point on the plot, if it is closer to another point, that means it's more similar in community composition. If they're farther apart, that means they're more different from each other. So these treatments did differ from each other, which is really interesting. And there's a couple of key pairings that I want to point out to you that I think really tell what is going on here. The first thing is that for both of the treatments where limpets were included in the plots, so the control in green and the acute in red, these two communities were different over the course of the experiment. So that inclusion of heat stress was changing something about the community. We also see, however, that it's not just heat that is driving this response, because the acute treatments and the acute with limpet exclusion treatments were also different from each other. So just because I was applying heat stress doesn't mean the community was responding in the same way. There's something about the inclusion of limpets in those plots that's driving that response. Interestingly enough, we didn't see that same effect of limpet exclusion if we just look at the controls, or the ones that were experiencing ambient treatment or temperature. So the control in green and then the limpet exclusion with no heat stress in blue were not statistically different from one another. So the inclusion or exclusion of limpets was also not the only thing driving this response. And then finally, just to really drive it home, if I exclude limpets, the control and acute are no longer statistically different from one another. So this relationship is only showing up that there is a response with heat stress if limpets are included, but limpets alone are also not necessarily changing the community. It's this combination of the two that's doing that. So to understand how the community is changing, I want to show you a tile graph where each of the columns is corresponding to one of the treatment groups. And again, these treatment groups are listed here on the left if you need that. And then each row corresponds to a species or a functional group of interest for us. Each of these cells is going to have a number in it, which corresponds to the average change in coverage over the course of the experiment. And the color is corresponding to the intensity of that. So a dark red is going to be an intense loss, and then a blue is going to be a strong gain or increase in their coverage. So if we put all that together, we see a few really interesting trends. The first of which is that barnacles and invertebrates generally, once again, did not care about me or my interests or what I did. There was no real change. All of them pretty steadily gained, and there was no difference between the treatments. Macroalgae, on the other hand, did not have that same response, and their response was really driven at a species level. So there were 3 macroalgal species, Fucus, Pelvetiopsis, and Endocladia, which you can see in this acute treatment with limpets included, had a higher level of coverage by the end of the experiment compared to the other treatments. And Endocladia especially is really indicative of this, because you can see in all the other plots, it had a pretty steady loss, but in this one, it was almost pretty negligible. You might be wondering, okay, they're all gaining in coverage. Is something being lost to account for that? Yes, Mastocarpus, which was the pretty dominant macroalgae in these plots, had a loss in just the acute plots compared to the other 3 treatments. So this seemingly dominant competitor was being negatively affected by the combination of heat stress and limpets, and then its other 3 macroalgal competitors were able to take its place and increase their coverage in turn, which is really interesting, at least to me. One final thing I want to show you about this experiment is one more NMDS chart, this time looking at the effect of time. So each color this time, instead of corresponding to a treatment, is going to be corresponding to a particular time point in my experiment. And there's 2 major things I want to show you with this. The first of this is that you'll see that the ovals for each time point are completely overlapping with each other. So time was not affecting this response, which is really interesting, despite the fact that I was going out there and continuously successively adding heat stress to this environment. There was no change to the response with the community. It wasn't intensifying the response that we were seeing. One thing, though, that is a little concerning is that time point 6 here in the blue was actually taken 1.5 months after the experiment ended. So with this only being a 3-month experiment and the heat stress being relatively low, this community response was still being maintained well after the experiment ended, after the last time I applied heat stress and the last time I excluded limpets, which is concerning when we think about it in the context of climate change, where now a very short-term heat stress is having effects that we see well after it is ended. So to sum up this chapter, how will the high intertidal respond to heat stress? Heat stress without limpets had no effect on the community. When we include limpets, heat stress specifically affected macroalgal abundance when they were present. And what we see is that Mastocarpus was really negatively affected by this combination, whereas 3 of its macroalgal competitors were positively affected. They were able to colonize that space. And we also see that while the effect didn't worsen over time, it did persist over time, which is maybe something to be concerned about. Okay, so wow, I'm doing good on time. Okay, great. So let me do some overarching conclusions for you based on all of this that I've talked about today. So to review really fast, in chapter 1, we saw that limpets are reducing algal abundance, but that this is really on these temporary communities, less so on the primary space occupiers, and that this response is primarily driven by Ladia digitalis, which is the most abundant species in the region. In chapter 2, we saw that 2 macroalgae, Pelvetiopsis and Masiella, are grazed seemingly regardless of conditions, but they are grazed more when they're heat stressed. Limpet species don't seem to differ in their grazing rates at an individual level, and limpets are not affected by the same heat stress that was causing the macroalgae to be more susceptible. And then finally, in chapter 3, we saw that heat stress is affecting community composition only when limpets are present. And specifically what we see is that one of the more dominant macroalgae, Mastocarpus, is reduced in abundance while 3 of its competitors are able to colonize that space. So what are some conclusions we can draw from this? We can see that digitalis is the most influential species, but that this is seemingly due to differences in its abundance or its non-consumptive effects, not its grazing rate, because we saw that that didn't differ. Limpets do seem to be able to graze on macroalgae, but these primary effects at a community level are not caused by their grazing response, especially when we think about Mastocarpus, which was really grazing resistant in chapter 2, but had the strongest negative effects due to limpets in chapter 3. We also see that heat stress increased macroalgal vulnerability to negative limpet effects, both consumptive and non-consumptive. They were more at risk during the grazing experiment and during these overall community trials that I saw in chapter 3. And then finally, we saw that community changes from heat stress seem to be persisting well after that heat stress has ended. So what are some future implications for this? We can predict that perhaps what we might see as climate change worsens and temperature stress becomes a more increasing phenomenon in this environment is that Fucus, Pelvetiopsis, and Endocladia may increase initially in abundance in response to heat stress as their competitor is taken out of the environment. However, what we see is that at least for 2 of these, we know that they are much less grazer resistant. Fucus and Pelvetiopsis was able to be grazed in chapter 2 in that grazing experiment, whereas Mastocarpus seemingly is not affected by grazing rates from limpets. So as heat stress continues to worsen and microalgae become less abundant, limpets may be able to switch to these 2 species and graze on them at a higher rate than they are currently. Overall, that means that climate change may be heavily altering macroalgal abundances, therefore altering our understanding of what constitutes the high zone community along the Oregon coast. And with that, I have to acknowledge some people. Yay! How much time do I have? No one knows. 10 minutes. Thank you, Gio. Gio's got it. Okay. Look at me. I'm chilling, guys. Okay. So first of all, I want to thank all of my funding sources. I had a whole lot of monetary support for these chapters, which I really appreciate. And I especially want to give a big thank you to the ARCS Foundation, which provided me with a scholar award for the first 3 years that I was here, which was really awesome to have. I also want to thank some people that really helped me out with these thesis chapters. Ellie and Nicole were my 2 undergrad mentees. They were really excellent. Can't thank them enough. I also want to thank three of Bruce's former students, Annette Olson, Peter Van Tamlin, and Gary Allison, who were really instrumental in helping me with designing these experiments, especially Annette Olson. I really had a lot of long conversations with her. I really appreciate that. Sarah Warden is another graduate student, not from OSU, but did really help me out with setting up some of these experiments. Chapter 2 would not be possible without Cinnamon Moffett and Trevor Erdman at Hatfield. They really kept me sane while I was driving out every day for an entire month. So I really appreciate them. And then I also want to thank Bruce McCune from Botany and Plant Pathology for his excellent class and for helping me out with a lot of my data analysis when I had my initial data. I want to really thank all of the members of my committee. So Felipe, Sarah, and Will, thank you for being here today. Thank you to Anna for when she was able to serve as my GCR, and thank you to Julia for stepping in when I needed someone to take that place. Thank you to Jane Lubchenko for being an excellent boss while I was able to work with you and just generally being such an amazing presence to have in the lab. It's really amazing to just hear everything that you've done and like hear your wisdom throughout the day. It's been great to have you back from the White House and be able to just like see you in the halls. So fun. And I also really want to thank Bruce, who has been the best advisor that I could possibly ask for. He's been such a delight. He really cultivates such an amazing lab community, and I can't imagine doing this journey with anyone else. So thank you. I also want to, whoa, we're good. I'm fine. I also really want to shout out the 22X teaching team who really helped me to develop as an educator and just generally as a person. You guys have been so awesome, and I'm so grateful to have been able to teach these classes with you throughout the years. I also want to thank my amazing labor union, the Coalition of Graduate Employees, for supporting me throughout my time as a graduate student and also giving me the opportunities to support others. It's been an incredible experience. I wouldn't change it for the world. I've learned so much from my time with CGE, so thank you. And then I have like 10,000 people in this list that I need to thank. Thank you to Sarah Hamilton, who I think is on Zoom today, for being such a good friend and such an amazing mentor. You were the first person from Corvallis I met, and you've been in my life ever since, so thank you so much. There's so many people that used to be in the Mengi lab that I need to thank, let me read them out right now. Brittany, Laurel, Zach, Jenna, Heather, John, Augie, Delaney, Laura, Saylor, Sydney, Chloe. You all are so awesome. Thank you for making such a great lab environment and being such good friends to me. Thank you, Leanne, in particular, for really dealing with all of my stupid questions that I have. And thank you also to Cindy, Catherine, and Gabrielle for also handling my stupid questions as well. Thank you to all of the IB office staff, Tracy, Teresa, Tara, Melissa, Rowan, for just keeping us all running correctly and keeping me in check when I need it. Thank you to so many other grad students from the department that I've met that I'm honored to call my friend. John, Risa, Cheyenne, Ricardo, and thank you to Drea and Maddie, who I met through CGE and I love so much. And thank you also to Lauren, Sarah, and Alexis, also from CGE. I'm so grateful to have met you all. And thank you to so many other people that I've probably forgotten. I'm so sorry. My brain is crazy right now. And thank you so much to all of my friends and family who have really supported me through all of the insanity that is grad school. Thank you to all the people that I met here. Sarah, who gave my excellent introduction, Megan, Audrey, both of my Emilys, Dan, Chip, Danielle. You all have been so excellent. I'm so glad that I got here just so I could meet you all. It's been great having you as friends. Thank you also to the people that I already had in my life who have supported me through all of this. Lily, Kat, and Evie, you all are so excellent. And thank you also to my wonderful mom in the top left for everything, for being my mom, all that kind of stuff. And then he gets his own slide. Thank you, Jake. You are the best partner I could have ever asked for. I feel like you have put in just as much effort into this as I have. I feel like you should be getting this degree with me. I love you so much. And with that, I don't know if I have time for questions. What time is it? I don't know. Oh, wow. Guys, I'm so time efficient. Yeah, Gio. Okay. Yeah. Hi, Lars. No, that's a really excellent question. As you probably know, the nutrient availability is different, especially between Cape Perpetua and Cape Foul weather. I haven't looked at it in particular, but I would imagine that at Cape Foul weather, there is just more macroalgae generally in the high zone. So that might influence the ability that limpets have to actually be able to graze down or affect these species compared to somewhere that's more invertebrate dominated like Yachats or Strawberry. Yeah. Yeah. Wow. I did so good. No one has questions. Yeah, Shine. Thank you. Yeah. Yeah. So before I set up that experiment, I did a few like field surveys to figure out what the average abundance is in like a 15 by 15 centimeter area. And that was what I used to set the abundances that I had for the limpets that were in the plots. Yeah. Yeah. Yeah, Megan. Yeah. I mean, I think it really depends, right? Because like depending on the season, the low tide is coming in at a different time. I mean, the thing with the high zone is that even a really weak low is still exposing a lot of these organisms, maybe not the entire high zone, but some of them, at least to aerial conditions in the afternoon, especially in the summer. I was able to get out there, so I know that, the sun probably is, but I do think that, yeah, that makes a difference. But I know that the high zone, at least because of its vertical location, is able to be exposed in the afternoon, at least for some amount of time, like an hour or so, like I was saying. Yeah. 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 I did. So at Fogarty, there was a bald eagle for a while that had a nest. Very cool, very patriotic moment. Yeah. Yeah, Alexis. NMDS, yeah.
Like, I know you said it was like a computer, you're trying to take all of these variables that you have and switch it out to something like
Mhm.
plot, two dimension. But I imagine there must be like many ways to do that. Maybe naively just like add them all together or something, for example. I guess I'm not sure how I should be interpreting those numbers. What is that actually?
Yes. Yeah. I think you and I will have to talk, but I will let you know. Don't you worry. Yeah, it runs a whole bunch of different like examples and figures out the best fit based on the data, basically. Um, but I'll teach you. Yeah. Okay.
All right.